Hello everyone, this is Jacob Pobio, the producer of The Apex. In this episode, Jan and RJ interviewed Distributed Bio's Chief Science Officer and Immunologist, Samson Yousef. They discuss her journey from Israel to Stanford and beyond, from couch surfing from when she first arrived to the US, to being a renowned immunologist working on treatments from everything from cancer to MS. She is now part of the team that is working non-stop to help fight the battle against COVID-19, testing billions of modified SARS antibodies in an effort to identify a treatment for COVID-19. Check out Distributed Bio here at www.distributedbio.com. If you want to support us, there are 3 ways to do so. One is to donate to our cause at www.patreon.com/theapexpodcast. Second, visit our merch line that is proudly partnered with Envision Clothing Company at envisionclothingcompany.com. The final one is completely free. All we ask is if you learn something from this episode or know someone that needs to hear our message, share it with them. Please subscribe and hope this pushes you toward your apex.
Welcome, Apex Chasers, to another episode of the Apex Podcast. As always, I am your host, Jan Almasi, and due to a massive pandemic in the United States, as of right now, I am not seated directly across the table from my co-host and partner in crime, Mr. RJ Holiday. However, he is here.
We're seated across the internet from one another in our respected quarantine areas.
Thank the Lord for the people at Squadcast, Libsyn, and the internet for being a thing. Otherwise, we would not be able to have this conversation right now. So how are things on your end, RJ? You surviving over there?
Good. You know, everybody's come over to steal my toilet paper and all that other good stuff. So I've been having to stand out front and keep the hordes away.
Oh, no, my Lord.
It's all been, like I said before, it's crazy the stuff that's going on around here. And it's lucky to have, really reliable sources and people being able to, step up and kind of reach out to community members who maybe don't have a way of finding out some of the prevalent information that's going on. And that's kind of what we're hoping to bring to the table with the podcasts and articles and blog posts and stuff that we're going to be putting out here in the next coming months, weeks, or whatever it may be, however long we're forced to be cooped up.
Right. Yeah, no, I agree. If you haven't followed the Apex Communications Facebook page or the group Apex Communications COVID-19, that entire purpose behind that was we wanted to build something where we are actively talking to people that are on the front lines doing research, people that are involved directly in this fight against COVID and all of these other diseases that we have, because we can't forget about all of the other things that we've been doing research on for years, just because we have a pandemic that's novel right now. And luckily for Apex, we've been able to have people volunteer to vet articles and make sure that people are having access to pertinent, correct data, rather than just sharing a Facebook post and it going viral and starting a massive rumor that is false. So there's a dedicated team of nurses, healthcare professionals, public officials, other people that are inside of that Facebook group on the Apex page that are making sure that the articles that we're posting there are correct. So with that, despite me and RJ being seated across the internet here local in Ohio, we also have somebody seated across the internet with us on the other side of the country in California. So Ms. Youssef, why don't you go ahead and introduce yourself to the Apex family?
Hi, everybody. Thanks for having me on your show. My name is Samson Yousef, and I am the Chief Science Officer at a biotech company called Distributed Bio, located in South San Francisco in the Bay Area. Do the research. It was a research on the floral and the tomato plants, how to expand. It was an agriculture, how to expand to make a smaller tomato with more fruits. And I fell in love with science, so I started helping the PhD students doing experiments. And I actually continued my master's there. I learned the molecular biology there. I made different type of libraries, but I made libraries that we fished new genes from the tomato plant. And then I was fascinated by immunology. So I moved to the medical school in the Technion, and I did my PhD in the field of multiple sclerosis.
So to back up just a little bit right there before we dive into the MS, for those people that are listening that may not understand what the word library means when you're using it in your context, why don't we explain what that is? Because I'm assuming right now there's a bunch of people wondering why you are looking at books on a shelf.
No.
Wrong library.
So it's like a library book, but like you can take a cell or a type of cells, isolate either the RNA or the DNA or the protein, and make a whole array of those compounds in a way that you can express them, but there's so many of them. It's like a library. You go pick a book. So if I want to find something, I fish in a biology method for that specific book. So that's why it's kind of like called like a library. So basically, it's just an accumulation of many targets in a shape of DNA. And so I go with my fishing boat and I fish whatever I want from that library.
Right. And it's a way to just kind of keep everything organized and to be able to pull out specific parts that you need when you need them.
Exactly.
Interesting. But I didn't know that it did stuff like that or categorize it like that. That's awesome.
I mean, yeah, kind of.
It's probably very painstaking.
Yeah, it sounds like it.
I wouldn't go into the complicated method how you fish the things in, but it's very similar with more sophisticated methods.
Yeah, I'm sure that it would 99% of that would go right over my head. So
everybody just use your imagination.
Yes.
Yeah.
So beyond that, so we are building these libraries and going through the program. So you said that you enjoyed the microbiology piece, and then you went into studying the actual immunology piece and focused on MS with that immunology studies.
Yes. So in that time, a lab in the medical school wanted a molecular biologist because they wanted to do some type of DNA vaccine towards some kind of molecules that affect the brain during multiple sclerosis, and they needed an expertise. I was not an immunologist. I learned immunology, but I wasn't an immunologist, so they needed a molecular biologist, and that's where my expertise were needed. So I went to that lab and did my graduate studies there, and I became an immunologist after 4 years of working every day in the lab. And human disease and in immunology fascinated me, and I wanted to continue to become a professor in academia. And so the classical thing is to go and apply and to do a postdoc, which is postdoctoral studies that you have to publish a very high record of peer review in order to be qualified to run your own lab. And so interestingly, in some way, I met a professor who used to be the mentor of my PhD professor. And we connected, and then I applied to his lab, and he accepted me, and I went to Stanford to do in his lab multiple sclerosis studies, and I worked and published many peer-reviewed papers on different aspects of I don't know if everybody knows what a T cell is, but it's one of our important cells in the immune system. And it's very important also to help getting, even like right now, people are talking about vaccine, and it's very important to help the B cells to generate antibodies after vaccine or after therapeutics or etc. So my focus was on metabolism of the T cells. So I used multiple different drugs, which is very funny because I worked on statins, and I figured out why statin could be beneficial in multiple sclerosis, and we completely dissected the pathway. But also I was always interested in, beside multiple sclerosis, in different diseases. So the nice thing is about Stanford, there's so many researchers and postdocs, and everybody wants to collaborate. So I worked with postdocs on using those small molecules in models for cancer, for graft versus host disease, and we kind of find a way how these molecules works in these diseases. And it's very fascinating because it's actually, although they are inhibitors for cholesterol, but in the immune cells, they have in cancer cells, they have a completely different function there.
Yeah, when you said statin, my first thought was like, oh man, I never thought that Lipitor would be,
actually, I worked
conjunction
on Lipitor.
with that Lipitor would be in conjunction with some type of MS or autoimmune disorders or anything. Because the running joke, and this is maybe a little bit in the healthcare realm, but we always say, oh, that's that life-saving Lipitor, that we have to wake a patient up for at 8:00 at night because they're sleeping to pass it because it's on their medical record that we need to pass that drug. But it'd be interesting to see, the look on a nurse's face when we're trying to critically think and saying, hey, we're taking care of this MS patient. Why they don't have high cholesterol levels? Why are they taking Lipitor? In the future, if that were something, to dive that down a little bit, because a lot of our audience is relatively well educated in the healthcare field and understand T cells and B cells and stuff like that. If we were to dive a little bit further into what that statin actually does to the cells inside of an autoimmune disease or cancer, what is a little bit kind of a lighter version of what they accomplish or what you've discovered?
Yeah, so absolutely. And so let's say in the concept of T cell, what happened is, okay, this molecule inhibits the early part of the pathway that makes cholesterol in our body, but that pathway is not only for cholesterol. It makes many more small molecules that affect, like Q10, for example, and there's other molecules, small molecules called geranyl or farnesyl, that actually their role is to anchor proteins to the cell membrane. So if something come from outside, to help negate the right signal to the cell, so the cell can produce the right material to fight or to absorb whatever comes towards it. Mm-hmm. So what the statin does in that way is it inhibits those molecules to be produced. And basically, when you have a big inflammation, tone down the type of inflammation, and the cells transiently stop responding or respond in a different way, because you skewed the pathway from option A to option B, and you kind of change your immune response to whatever you are responding against. And so wow. and we actually proved it even in a model, in an animal model, that it's transient. So that's why that's what our reason was you need to take it every day, because that inhibition can go back. And so in order to keep it consistent, so you need to take it every day, because that inhibition takes, in our hand, was like 8 hours. So before it switched back. So wow. It was actually, it took us a while to figure it out, because it was very puzzling, because in the culture it was really easy to do it, but like we had to prove it, like why this change happened to the T cell, what's going on there. So and the good thing is, like, the best thing for scientists that when you do something, some other lab would do the same thing and prove that you're right. So we had a lot of papers that showed similar things. And it's so funny that you're mentioning this, because we also worked on reducing using those molecules into the tumor model, a lab approach at Stanford, and we did those studies and it worked. And in there, it was mainly basically on like killing those tumor cells. It was like it affected pathway, because those small molecules affect many proteins in our cells. So there's multiple or pleiotropic or many effects that could happen. And I was browsing LinkedIn last week, and I found a paper that's saying, statin inhibits through this specific pathway killing tumor cells. And I immediately emailed my mentor, Larry Steinman at Stanford, and it's like, Larry, look, a decade and a half ago, and they're still repeating our study. So I actually think it was very, very funny. It couldn't be more funny than
That's
this.
awesome. That's so flattering. And then also, well, I mean, and my expression was probably pretty much key to like everybody that's going to listen to this episode. At least the nurses that I know that are going to listen to this are going to be like, wait a second, statins? Really? Out of all drug classes, that's the one?
I mean, we used to laugh in the lab and say, we should put statin in the water and just drink it in a low dose.
At all times.
Yeah. Okay. So this is all at Stanford, right? This is where all this research is going on?
Yes, that was all at Stanford.
Do you have anything to talk about at Stanford there, RJ, or?
Well, the main thing that I heard you say is because you were able to go to Stanford was it a graduate program, you said? Postgraduate. You were, because like the labs there and everybody's there was working on many different things, you were able to kind of put your work to several different fields?
Yes, because everybody wants to collaborate. There are so many postgraduates and graduates in many programs in these big academic institutes that like collaboration is very encouraged, and there's a lot of like collaborative work that can initiate, like even from those, a few small initiative trials were initiated just because of these collaborations.
Gotcha. That's extremely interesting. Is it not usually like that? I mean, just maybe in bigger academic settings like that, or is it mostly like is there certain places where like if you're going here, you're going to be working on this and that's it? Or if you go to another place, this is what you're going to be working on day in and day out with no other exposure to any other, fields or scientific work?
I don't know. I think it depends on how big the institution and how many people are coming from where and what. And like in my PhD program, I collaborated with groups around me in my department, but we weren't big enough to like go and initiate a collaboration outside of the group.
Gotcha.
So yeah, it depends on how large. Like I would say, like in institutions like Stanford, Harvard, Yale, Princeton, this happens all the time, and I'm pretty sure in other countries. But I can't say because I wasn't in. But big universities like Oxford or like all these huge amount of, when you have a huge amount of researchers and labs and people coming from all over the world to work. In Israel, I would say like the Weizmann, nowadays also the Technion, I'm talking about like 20 years ago. Now it's way more, and the communication is happening because of also the social media. Now you don't have to be physically there. Like we used to check our email once a day. Now like you check your email once a minute, right? So
yeah.
Right.
So also like the social media brought more collaboration. So I think now it's everywhere.
Awesome. Was when you moved out here, was that your first exposure to the US?
I had family here, so I visited a few times. And when I came to interview also I visited, but living outside my country, yes, it was my first time.
Were you nervous?
Oh, you want to hear the truth?
Yeah, let's hear it.
I think I cried every day for a year.
Oh man, you know what? So I was not alive for that portion of my mother's life, so my mom immigrated here in '93, and then so I'm really I'm going to go back to the Slovak embassy when all of this is over and reestablish my dual citizenship. But half of my family still lives in Košice, Slovakia, and my mom immigrated here in '93. And the way that my dad explains their dynamic of their marriage for the first year was essentially the same thing. Her locking herself in her room and crying like every day because she couldn't. There was so many, the homesickness, the dealing with language issues that she was going through, and just a whole bunch of different things that compile. So anytime that there's that crossover from country to country and going into a new place, I really feel super connected to that type of experience, because although I didn't personally go through it, I still, to this day have my mom break down every now and then because her mom and dad are still in Slovakia, and what that distance can feel like, especially if you still have family overseas.
Yeah, absolutely. I mean, it's also the cultural shock, because it's like, how do I behave? What do I do? How can I find an apartment? And how can I do this? How can I buy a car? I don't, like all these emotions, and working in the lab, and I need to put my research from zero, and they're like, you're so independent, your boss give you a project and you have to run it by yourself. There's nobody who can help you. And on top of all of this, and I came in 2000, in the peak of the .com, and I for 3 months I could not find an apartment because I didn't have credit history. And I was like between couches, between friends. It was quite a journey, but it shaped me. It made me who I am. So I wouldn't change it.
And there it is, right there.
Yeah.
That is the people that I've come into contact with to either take one of two pathways when they experience something like that. And it seems to be a theme with the type of guests that we have on the Apex specifically too, is that you can either look at it as a woe is me, that happened, have pity kind of thought process, or like you just said, it shaped me into who I am. And these are the lessons that I learned in those situations. That, I mean, being couch surfing and not having an apartment for 3 months because of no credit history probably makes it extremely easy to appreciate having an apartment or any type of simple thing like that, where other people may overlook that.
Oh yeah. I mean, I was lucky because Stanford has a rule that in the bank, they will give you, regardless of your credit history, they will give you a Visa or a debit card. So I could build my history, and they helped me get a loan to buy a car.
Wow.
But in the beginning, like, you're on your own, and I have no family in the Bay Area until last year. My cousin, who also came from Israel, and she has her family in New York, she relocated. So that's the first time that I have a family in the Bay Area, like really close family. All my family I have 2 uncles in the East Coast, 1 uncle in Canada, and all my siblings and my family is in Israel. So I was like almost 19, 18 years living by myself, and my family was my friends and my colleagues. So it's quite the journey, I would say.
Yeah, no kidding.
So when we're moving from Stanford and beyond that, you ended up, is that when the transition happened between Stanford and then moving to Pfizer right off the bat?
Yes. So go ahead.
Well, I was going to say, so at Pfizer, there was this, I believe, and you can correct me if I'm wrong on this, just through the research that I was trying to do prior to the interview, this protocol B8011001 that was the study that it looks like is listed on your distributed bio, and the calculation for melanoma and ovarian sarcoma cancers.
The anti immune drug, a checkpoint inhibitor that last year I believe 2 years ago they won the Nobel Prize on these pathway, the CTLA4 and the PD1 in medicine, or I believe. I moved to Pfizer in 2008, and that's another story because I stayed long enough in the academia to have my credit to become a professor, and I had grants that I can open my own lab, but it was like the recession and there was no academic jobs that fits my expertise. And after 2 years of long search and applying and academic job, because NIH funding were not available all over the world, I tried, and I told my boss, I'm done. I give up. I don't seem to have an opportunity to become a professor, so I'm gonna try my luck in the industry. And actually it was funny because after all these job interviews that are really draining and I got 3 offers, but 2 of them were shut down because no funding at the end of the university, and 2 came late after I actually started working at Pfizer. So actually looking into the industry, like almost every interview in every company I got a job. So I was like, okay, I had like discussion with my mentor, my PhD mentor, and my uncle who is a scientist in industry, and they told me it's good to start your career in a big pharma or a big company, so you can learn all the expertise and all the multidisciplinary. Yeah. And so I went there, and this was a site that was bought a few years ago. It was a neuroscience site, and they needed like a lot of immunologists to move it to an immunology site for antibody development. So I moved there. I believe one of the first immunologists to join that small site in South San Francisco. And I started building helping build these essays and develop multiple programs with them that are related to MS and lupus and arthritis, but those are confidential for IP. I can't talk about them because they're still going. But then when the hype of the immuno-oncology happened, and they found that these drugs that you basically, when you attack you make your immune system attack the tumor with these antibodies. They call it checkpoints, because basically those molecules stop the T cell and the B cell from working in our body to fight the tumor. So those molecules happen to stop this pathway and make those T cells become even actually the opposite from autoimmunity and inflammation. We want to make more inflammation to kill the tumor cells. And they told me, okay, you are going to lead the efforts for the internal PD1 program, which is the program that you just mentioned. And so I took an antibody that was generated in a hybridoma in a mouse and moved it through humanization, meaning make it more suitable to the human so it won't make side effects, and move it through phase 1, almost close to phase 2 before I left, from the bench to the bedside. And I'm very humbled that our trial, at least phase 1, like just knowing those patients that, because I don't know if you want me to elaborate on what phase 1 in clinical trial is.
No, but yeah, for the people that, like I said, a good portion of our audience are medical professionals, but there is a good section that are not. So anything that you feel like you can elaborate on is better for the listeners.
Yeah. So basically, phase 1 is you have certain criteria, you build certain criteria, and those people who go to phase 1 is people who, like, this is their last hope. Nothing works for them, and they needed very, very severely something that helps them. So so we did the phase 1, and we were like very happy to see that our drug was tolerable, that we had responses in patients. And for me, that was very humbling. It was very, very humbling, because I kind of touched what it means to be really affecting human life. And
you got the chance to be at the bedside. Yeah.
And to me, it was worth every academic position I missed. I wouldn't change. I wouldn't change anything that happened. Everything happens for a reason, I guess. And it taught me different part of this whole human health, because in academia I learned how to do basic research, write papers, write grants, get grants. Here I learned how to prepare a drug for a clinic, how to write those filing that you file for the FDA. How to write a protocol to help the clinician to write a protocol for a clinical trial. What is a protocol for a clinical trial? So it's kind of like looped me in a different area that is really close to human health, which I love very much.
So now you got the chance to directly interact with these patients that were in the phase 1 trials?
No, we cannot, just out of respect to the privacy. You just what happened is that you connect with the centers who run the clinical trial, and they have so I was the research lead, is the lead on the program for research, and there is another colleague that is the MD who leads the clinical program, and together we lead the program together. And what we do is we meet on a weekly or monthly basis so there are centers that are running these studies, and it's very, very like organized and controlled. And so all the data, the clinicians, so we have like discussion with the clinician that lead each clinical site, but we do not interact with the patients. That's not in the protocol. We cannot. So and everything is blinded. We don't know who the patients are. It's all numbers. They go to, like, everything is organized. All the data goes into center, and the center organize, clean up the data, send it back to the site. And like, so we do not know who, and we do not know who is the patient and what's their symptom and who they are. We don't have this information. It's just numbers for us, just because of protocols and IRBs and reviews and privacy.
And non-biased data and all that other kind of
absolutely.
stuff. Now, and that's where it kind of I always find it interesting, and I'll use kind of residents, I guess, as an example of this. Just being an RN and my training leading all the way through nursing school was kind of half and half, the science and then in-person clinical orientation and interacting with patients. And I've always thought that it was very intriguing when a new resident physician comes into the hospital, how much knowledge they have when it comes to the chemistry background, the microbiology background, the pharmacology background, but then watching them attempt to interact with these patients in these like critical moments or these very touchy kind of places where it may be somebody that they're about to go into a surgery that's their last hope, or they're actively having a heart attack and they need to go in and do this, or they just got diagnosed with cancer. And in the back of my mind, as you were talking about all of these people that this is their last hope and they're going through this clinical trial and how it affected you seeing the positive results of that clinical trial, I couldn't help but picture myself as that RN doing, whether or not I knew I was a part of this clinical study or not, or if this patient was in a trial, but working at one of those centers and what that would be like to realize that this is this person's kind of last hope. And that it ended up statistically successful is a phenomenal thing. Even just the numbers without the patient interaction, and just knowing that that's the situation that those people are in and that you're actively trying to do something to help them is one of the most rewarding feelings in the world.
Yeah, absolutely. I cannot emphasize that enough. I know it might be selfish, but it's really humbling to me to be part of it. Not in a selfish way, but it's just like, oh my God,
You're just like in awe almost that you got the chance to help.
Yeah.
And I love that. And I actually find that more with people that are not from the US. And I say that with love to all of my people that are born and raised strictly in America and have never really experienced overseas. But I know my family in Košice, they're so focused on this that, like you said right there, that proud humility almost, where it's kind of this weird mixture where it's like, yes, we're very proud of what we did, but we also want to make sure that we're maintaining that humility. Where some of the friends that I've interacted with stateside are very forward about, oh, I was a part of this, like getting super excited. Look at me, look at this piece that I'm a part of. But I love that feeling that I'm getting just from how you can tell how passionate you are about what you do, and that you're doing it for the purpose of helping others and being passionate about loving what you do on a daily basis, not because you're chasing the glory of curing a bunch of people. Mm. And that's a beautiful thing to see.
Thank you for that. I met a lot of people that feels the same way in my life, but also it's very hard for me to explain it, but it actually makes you look at life in a completely different way, because all these other nonsense doesn't matter anymore. Like those people really, maybe they weren't all cured, but extended their life for having a Christmas with their family or holiday with the family. That puts you like, you don't care about if you win the lottery, or like, for me it was like a completely life-changing experience, I would say. Made me feel very humble.
Yeah, I'm sure. It like you said, it opens up. Life is all about perspectives.
Yes.
And especially with, I feel like that'll kind of lead us kind of directly into all of the shenanigans that are going on in the world right now.
Oh boy.
With perspective, I think that's actually the perfect word to try to transition us into everything else that's going on. But lenses are huge. There's this poem that I read on a daily basis called The Desiderata by Max Ehrmann. And it was discovered on the wall of a church, I believe, and was uncredited for a while, but they ended up finding out who wrote it. But the very last line of that poem says, with all of its sham, drudgery, and broken dreams, it is still a beautiful world. Strive to be happy. Like, be gentle with yourself and strive to be happy. And that last line, just to me, is inevitably, there's this cliche of trying to find the silver lining in things. But with all of the other stuff, once you've experienced that type of experience that you're talking about, or that I compared to the bedside, or like a first responder going into a house fire, or a police officer, or somebody in the military, or even just somebody that is a regular job and they experience something that throws them out of their normal loop and forces them to look at life in a different perspective. The entire world is experiencing that right now.
Yes.
They're being pushed out of their comfort boxes and being forced to look at the world in a completely new perspective. And it's, I'm curious as to what your perception is on it, with seeing what people are doing and how humanity is reacting. What is your take on how people are dealing with being pushed out of their boxes right now?
So, okay, I would say yes, people in some way are panicking because this is something probably some part of the people never experienced any massive incidents, like, for example, 9/11 or something. So for them, this is something new. They just heard about it in the past, or they never experienced some world change, whether in disease, pandemic, or war, or crisis. So for those people, I would say just follow the common sense and take care of yourself. and don't be afraid. Don't be be cautious, be aware, do what everybody's in the news, the health professionals that are advising us to do. And there is the other people that a lot of people in our company, because we have a program to help this COVID-19, they're like willing to come and put their health on. I mean, we are all trying to be safe, but this is like type of enemy that we can't even see, right? So we don't know if it's in the room and just so excited. I'm also overwhelmed of how many people in many places, not only in our company, that overjoyed just to go and want to help find some cure or some medicine and work on helping patients and in the science and in the healthcare industry. So it's very stressful but very humbling as well. So
yeah, I've kind of gotten the same perception. RJ, I'm curious about what you've seen as well, because I know me personally, just being in the healthcare field and everything else, when we were all kind of told, hey, this is what's going on, this is what we need to do, pretty much everybody was willing to volunteer either OT, overtime, or, the mentality is, okay, we're going to roll up our sleeves and we're going to get it done. It's like coming from a military background to see healthcare professionals take that same mentality where it's almost like we took an oath and we took a promise to make sure that we were going to take care of people. And that's what we're going to do. Has been a beautiful thing to watch. But there's been people like sports athletes donating their salaries to pay people's
the arena workers
bills
and stuff. Yeah.
I mean, and RJ, so like I kind of want you to explore that avenue. I know I can see it from the healthcare side, but what have you seen as far as like society in general and people really reaching out to each other and being humane?
It's different for like everybody, because if you do get most of your exposure to what's going on outside of like what CNN and stuff is talking about, nobody posts about any of the good stuff. I hear horror stories of, family friends. The lady tried to buy 6 loaves of bread, didn't want to give up one until, one of the cashiers is like, you can't even buy 6 loaves of bread. They won't even let you. So put it back.
Right.
Which is sad to hear, but then you hear a lot of other stories about, locally ran businesses and stuff that are doing everything that they can for, there's, what was it? I just read that some landowners were telling the companies that, for the extent of this is going on, to use their rent money to pay their employees, whatever that could be, since they're unable to work or can't go in there and work and stuff like that. It more or less, when it comes to, I believe, when it comes to your average everyday person, the little stuff that your neighbor, your friends, your family and stuff can do for the people that surround them is going to be what helps out them the most. Helping, like, I think, what was it? My girlfriend's family, they have an elderly neighbor or whatever, that lives right to the right of them. And instead of making them go out and risk exposure to whatever might be going on, I think Steph's mom agreed to just do her grocery shopping for us. And she was going out there anyway and would be able to get what, I mean, that's in that little heartwarming stuff that, you don't usually see most people going out of their way to help one another. Yeah, the grandiose things where billionaires are helping out their employees and paying people salaries for that kind of stuff. But it's nice to see individuals going around and helping, where they can. And the little minute stuff that people, take for granted now, can't leave my house, can't do this, can't go, get my prescription, whatever that. If you're willing and able to help someone who is either, less fortunate or is putting their own health or family's health on the line to do stuff that you're already going to do or be there, I would highly suggest you step up to the plate and do what you can for the people that surround you and see you every day. It's funny to see the obviously anybody who listens to the show knows I'm a big gym rat, so having the gym shut down is a little upsetting to me, but the gym community has come up with some very interesting at-home workout routines that all I see is just being shared all over the internet, so that people know who run their life heavily revolves around physical fitness and that kind of stuff are able to spread that type of information and keep people from going stir crazy in their own homes here over the next month or whatever it may be.
Yeah, those little acts of humanity have been amazing to watch come forth, and I think that's a perfect call to action to everybody, is to say, hey, it's not there's only so many billionaires in the United States that are going to be able to pay everybody's salary for a certain period of time. Mm-hmm. But we all have a neighbor, we all have somebody that we can reach out and lend a helping hand to, and that's what's going to be important. But so with all of the research and everything, we've kind of led into landing at Distributed Bio. You mentioned prior to the mics turning on that you met Mr. Glanville at Pfizer and then transferred over into Distributed Bio. So why don't we talk about what that transition looked like, meeting Jake and coming over to Distributed Bio, and then what you're currently working on at DBio when it comes to battling the COVID-19 pandemic.
Sure. So I met Jake in 2008. We joined Pfizer at the same time. So I don't know, we had some discussion or he heard me speak in the research updates or whatever in immunology, and he as part of his bachelor degree, he was always interested, and he did molecular biology, I believe, but then he was very interested in the immune repertoire, or in like antibodies, as you can say, he's an immunoengineer, antibodies profiling, and he built a lot of technologies around there. So we actually collaborated together on some projects, and then by that time he felt that he wants to move forward and get his PhD in immunoengineering, and so and he started Distributed Bio in 2012, but we stayed in contact through the years, through our friendship, and he always mentioned, when I need an immunologist, I'm going to call you, Sawsan.
You're just waiting for the day.
And I kind of rolled with it,
Right.
And so when time happened, it was like a good transition, because I already had accomplished what I needed to accomplish, and it just happened organically that they were ready to have me join Distributed Bio and help setting up functional assays for many program, and functional and also not only functional assays, but also different technologies that rely on cell biology and immunology, like for example finding antibodies against difficult targets that you need to do the fishing boat through cells and building these systems. And also, like I'm pretty sure, since you're in the medical field, know about CAR T. So we're building a platform to find functional antibody through the CAR T itself. So I'm spearheading this program, and I also I was doing one type of experiment, and I found something that I kind of didn't expect, a result I didn't expect. So I came to Jake and I'm like, Jake, I think we should look into that type of immune therapy for cancers that do not respond to classical checkpoint inhibitors. What's going on? And what I like about this company and Jake is although the ideas might sound crazy, if you convince, he would see the vision that I have. So we kind of in a parallel line of visions on how to be innovative and how to find. So I have another program that is still evolving, so I can't talk about very much, but hopefully in the future, once we finish the proof in vivo in the animal models, we can talk more about that. Could help many patients who in cancer, hopefully, that don't have like the right therapy right now.
Right.
So,
and
yeah,
that was something that me and Jake we talked about when we were on our interview was that ability to see somebody's vision with them, not for them.
yes.
and supporting those visions. And it's kind of the same between me, RJ, and Jacob, our third co-founder here at Apex. It was, not a whole lot of people in small town Northeast Ohio that are pursuing podcasting as a full-time gig. But luckily enough, we were all able to kind of land in the same place at the same time and support each other and see each other's visions to push it to the point where, now we're sitting and doing an interview with you across the country. It's a beautiful thing when you find that group of people that's able to support you and push your dream into becoming a reality.
Yeah, I would say me and him really, our vision kind of like parallelly and cross. They always like, that's what I like about distributed bio, that we have these innovators and visionaries like Jake, and just we understand each other scientifically and appreciate it and just roll with it and no fear.
So what? Go ahead.
And so just going to the COVID-19, so I'm pretty sure Jake talked to you about what our effort here of finding new antibody, like from old antibodies against the other virus, the SARS virus, just modify it. So what my involvement is I'm gonna, because I know how to like build those functional assays very quickly. So I'm trying to build these functional assays that can help quickly once they finish finalizing those antibodies, we can quickly test them to see their functionality on cells and choose the best candidates to move forward very fast. So that's why I've been working in these shelter-in-place days to try expedite as much as I can with this program.
Now, when you say very fast, what does that timeline look like for the general public?
To build these assays? To build the assays,
yeah.
I would say 2 to 3 weeks.
Right. And so that's the kind of thing that I kind of want to address a little bit of what that process looks like with you guys on the front lines, because there's a lot of people that I talk to that I know that there's a Canadian company that just released data on like potential vaccinations and other stuff like that. But what this timeline research actually looks like as far as the vaccine versus antibody therapies, and that very fast for somebody that's extremely experienced in immunology and building these functional assays still means 2 to 3 weeks at a minimum to even get started testing with the functional assays.
So okay, so let me just touch base quickly on antibody versus vaccine. So vaccine, it takes, even in this speed, it takes to realize if the vaccine is working, it takes between a year to a year and 8 months, because you still need to educate your immune system to generate those antibodies to block the virus. So that will be in the future, hopefully in prevention mode, or if this, hopefully not, if this pandemic stay long, then maybe the vaccine will help. Right now, we need like a cure. We need an antibody that will stop the virus from doing what's doing. So the immediate therapy should be like either a small molecule, like they've been trying the small molecules from the drugs from Gilead and from other companies like Sanofi, to see if they can inhibit the infection, but also a lot of companies working on developing antibody that will basically abolish or neutralize the function of the antibodies. And as Jake mentioned, we will have those antibodies ready in early April. And so I'm building that parallel assay to like immediately check, so we can make sure our antibodies are safe, make sure our antibodies will not attack our immune system, and all kind of important assays that will help provide the fastest way to our antibody to move forward, our drugs to move forward.
Mm-hmm. Yeah, and that timeline, I think that and I've been trying to do some of that education too through the posting and everything else that I do, is that there is a big difference between vaccine and antibody, and that so many people, I think the general public is just so used to hearing that word vaccine that anytime anything hits the headline and it's vaccine oriented, they're like, oh my gosh, like it's only, it's still a year away. We're all going to die by then. And that hysteria kind of kicks in where it's, okay, we're maybe blowing this just a tiny bit out of proportion. But the antibody research is really what I'm interested in and focused on, which is why I was so excited to talk to you today, because of your direct involvement with that, and knocking that out. So why don't we kind of walk somebody through the process so you build the initial functional assay, and now you're doing tests on these. First, how many of those are you building out right now?
You mean the functional assays or the antibodies?
The the different antibody types that you're testing. So
what we are generating is we are taking those antibodies that we know are they are neutralizing for the cousin virus of the SARS.
Right.
And so Jake with his genius built these libraries, which means like he made many versions of these type of antibodies. We made many versions, billions of versions of these original antibodies with different alterations, so we can find and nail some that will be specific for the virus of the COVID-19, which is called SARS-CoV-2. So, right.
So we're dealing with billions of different possibilities.
Yeah, and so it's like I said, in a library. And so what we're gonna do is take those binding domains or these proteins that the virus uses to attack those cells in the lung or in the kidney or wherever there is that receptor on our cells in the body. And we're gonna take that protein and basically fish those strong antibodies that will block this interaction with the cells. And those antibodies are gonna be our lead antibodies to move forward. We only need one good one, but we're making billions, and we are fishing those antibodies. And then the cell assay. I mean, I'm doing it in parallel, but Jake is not. He has like these collaborators in the army, and that they're gonna do their functional assays also on neutralizing the virus. So we will know early April for our success. So
and then moving into the next phase, once you identify and narrow it down, the next phase of that process.
Then we're gonna take it expedite to generate those antibodies. And Jake is talking to many companies or with the army as well, how to take it in a fast track to GMP to put it in patients as soon as possible.
Right. Yeah, it'll be very very interesting, and to keep in contact and to continue to push out this information from basically just like I said, being on the front lines. I love having these types of conversations just because I'm getting off of this recording, and in 20 minutes from now I'm going to be on my way to work in the ER tonight. So being out and feeling like I'm on the front lines actively, like trying to triage patients or take care of regular ER admissions on top of dealing with the pandemic and educating and everything, the more knowledge that we're able to put out to our listeners and the world in general, with the internet as a thing, since we're all cooped up and we have nothing else to do, we might as well educate ourselves on what's going on so that we can continue to assist. And then when stuff like this happens with distributed bio that's pushing forward, if there's any people that are listening to this right now, I know Jake told me to talk about this as much as I could, that are in the biotech industry, that are with backgrounds in immunology or backgrounds in this type of research that would like to try to collaborate with distributed bio, shoot me a message. I'll leave my email in the show description of this show, and you can I'll get you in contact with Jake and the team at Distributed Bio so that you guys can start coordinating, which is something that I love and I absolutely 110% respect about your guys' organization, is just the amount of collaboration that you're willing to do outside of obviously protecting your intellectual property. But when it comes down to the rubber meeting the road, I feel like Distributed Bio is very, very revolutionary and innovative when it comes to collaborating with people.
Yeah, and I want to say this is here we are dealing with something really urgent. It's not about any information is very important. Any collaborative information or any knowledge of any scientist in the world is very important, even if it's coming from a pharma, from a biotech, because right now it's not about the money and how much money we're going to make. It's about humanity. And this is really, really important. And this is what I like about our founders and Jake and Distributed Bio, because right now we're dealing with a humanitarian crisis and we all need to put our hands together to solve it. Right now, it's really important to do that. And I have to say, I'm very humbled with our team here, how much they are working so hard on getting the job done as soon as possible. And I'm very proud to be part of that team. It's another experience, humble experience that I'm experiencing. I feel I'm so lucky, and I hope that we can help contribute to this pandemic. But regardless, any result is very important right now. It's not about who is gonna race to the finish line. It's about we need to race to the finish line, all of us.
Right. Yeah, making sure that we all get there in one piece.
Exactly.
Yeah.
So please wash your hand, and if you don't need to go out, stay safe, stay home, and let's minimize interaction so we can completely help while we are developing these drugs.
Yeah, I agree. Well, with that, I mean, RJ, do you have any last comments or anything there? But I think
we covered it all. Just everybody do your little parts where you can for the people who can't for themselves, and like she said, wash your damn hands.
Well, I have to say, this has been a pleasure. I definitely appreciate, especially with all of the stuff that you guys have going on with the shelter in place and working on these extremely important initiatives, to take the time to sit down and have this conversation with me and RJ today.
Yeah, absolutely. We appreciate it.
And we've made this expression to Jake and Sarah and other people on your team that are now emailing me and talking about collaborating, that anything that we can do to make sure that we're pushing out as much information for Distributed Bio as possible is what we're aiming to do here at Apex. So I consider you guys definitely a part of the family at this point.
Mm-hmm.
I'm humbled and honored to join your podcast, and we're doing our best. And please forward any information to us. We're happy to talk to anybody.
Absolutely.
Beautiful. All right. Well, with that, everybody, again, this has been Samson Yousef, the Chief Scientific Officer for Distributed Bio. Her information will be inside of the show description. And make sure that you check them out along with the Netflix documentary Pandemic that talks about a lot of the research on the Centavax project that Distributed Bio has done. And until next time, wash your damn hands.